Researchers from the University of Minnesota Twin Cities College of Science and Engineering and Medical School have developed a unique head-mounted mini-microscope device that allows them to image complex brain functions of freely moving mice in real time over a period of more than 300 days. The device, known as the mini-MScope, offers an important new tool for studying how neural activity from multiple regions of the outer part of the brain, called the cortex, contribute to behavior, cognition and perception. The groundbreaking study provides new insight into fundamental research that could improve human brain conditions such as concussions, autism, Alzheimer's, and Parkinson's disease, as well as better understanding the brain's role in addiction. The research was published today in the peer-reviewed journal Nature Methods. The study authors will also present their research at the virtual 2021 OSA Biophotonics Congress, Optics in the Life Sciences on Thursday, April 15. In the past, scientists have studied how neural activity in specific regions of the brain's cortex contribute to behavior, but it has been difficult to study activity from multiple cortical regions at once. For mice, even the simple task of moving a single whisker in response to a stimulus involves processing information in several cortical areas. Mice are often used to study the brain because they have many of the same brain structures and connectivity as humans. This device enables us to image most of the mouse's brain during free and unrestrained behaviors, whereas previous mesoscale imaging was usually done in immobile mice using devices like the MRI or two-photon microscopes, said Suhasa Kodandaramaya, the senior author of the study and University of M. This new device allows us to understand how different areas of the brain interact during complex behaviors where multiple areas of the brain are working together simultaneously. This opens up research into understanding how connectivity changes in diseased states, traumatic brain injury or addiction. The new mini-MScope is a fluorescence microscope that can image an area about 10 mm by 12 mm and weighs about 3 grams. This allows holistic imaging of much of the mouse brain surface. The device is used for calcium imaging, a technique commonly used to monitor the electrical activity of the brain. The device mounted on the mouse's head captures images at near cellular level, making it possible to study connections between regions across the cortex. The researchers created the miniaturized microscope using LEDs for illumination, miniature lenses for focusing and a complementary metal oxide semiconductor, CMOS, for capturing images. It includes interlocking magnets that let it be easily affixed to structurally realistic 3D printed transparent polymer skulls, known as C-shells, that the researchers developed in previous studies. When implanted into mice, the C-shells create a window through which long-term microscopy can be performed. The new microscope can capture the brain activity of mice for almost a year. The researchers demonstrated the mini-MScope by using it to image mouse brain activity in response to a visual stimulus to the eye, a vibrational stimulus to the hind limb and a somatosensory stimulus presented to the whisker. They also created functional connectivity maps of the brain as a mouse wearing the head-mounted microscope interacted with another mouse. They saw that intracortical connectivity increased when the mouse engaged in social behaviors with the other mouse. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.